Freeze drying, also known as lyophilization, is a vital process in the production of many pharmaceuticals, diagnostics, and other life science products. While the process is highly effective when performed correctly, there are many misconceptions about freeze drying, which can compromise the quality and efficacy of the end product. Misconception: freeze drying is a straightforward procedure. Reality: freeze drying is a multifaceted process that extends beyond simple freezing and drying. It entails intricate steps, requiring specialized equipment and expertise. Freeze drying encompasses several stages, such as freezing, primary drying, sublimation, and secondary drying, desorption, each requiring precise management of temperature, pressure, and time to achieve optimal results. Additionally, the composition and properties of the product being freeze-dried contribute to the process's complexity. Choosing appropriate equipment and possessing expertise are crucial in freeze drying. Specialized equipment is indispensable for creating controlled environments and accommodating diverse product types. Without the correct equipment and knowledge, attaining successful freeze drying becomes a daunting task. Comprehending heat transfer principles and the involved materials is vital. Factors like container selection, heat transfer dynamics, and suitable materials significantly influence the freeze drying process. Misconception: all freeze drying equipment is the same. Reality: actually, freeze drying equipment is not all the same. There are various factors that differentiate freeze drying equipment, including design, capacity, functionality, and features. Here are some key points to consider when comparing different freeze drying equipment. 1. Scale: freeze drying equipment comes in different sizes and capacities, ranging from small laboratory scale units to large production scale systems. The scale of the equipment determines the quantity of product that can be processed in each batch. 2. Configuration: freeze drying equipment can have different configurations, such as shelf freeze dryers, rotary freeze dryers, or manifold freeze dryers. Shelf freeze dryers can have either internal or external condensers and may be built for steam sterilizability. Each configuration offers distinct advantages and is suitable for specific applications or product types. 3. Control and automation: the level of control and automation can vary among freeze drying equipment. Advanced systems may provide precise control over parameters like temperature, pressure, and drying time, along with automated processes, data logging, and batch reporting capabilities. 4. Vacuum system: the type and efficiency of the vacuum system used in freeze drying equipment can differ. A robust vacuum system is crucial for creating and maintaining the necessary vacuum conditions during the freeze drying process. 5. Refrigeration and cooling systems: the cooling system in freeze drying equipment is responsible for cooling the shelves or the product itself and for condensing the vapors that are being removed from the product. The temperature range of systems varies depending on the condensing rate requirements and type of solvents being condensed. The efficiency and cooling capacity of the refrigeration system can vary, impacting freezing and drying rates. 6. Shelf design: the design and material of the shelves used in shelf freeze dryers can vary. Some systems offer heat only. More sophisticated freeze dryers offer fluid-filled and cooled shelves, allowing the product to be frozen inside the freeze dryer. This feature provides uniform controlled freezing of the product for proper process control. 7. Additional equipment: additional equipment like process analytical instrumentation, capacitance manometers, isolation valves, dry vacuum pumps, and other devices be provided to improve the overall efficiency and effectiveness of the freeze drying process. Selecting the appropriate freeze drying equipment features is crucial for achieving optimal results. Factors such as product requirements, batch size, and available resources should be considered when making equipment choices. Customization and flexibility are significant factors in freeze drying. Different applications may require specific features like temperature and pressure control, adjustable drying times, or additional functionalities such as clean-in-place (CIP) systems. Having equipment that can be tailored to meet specific needs ensures efficient and effective freeze drying processes. Scalability is another important consideration. Pilot or small-scale operations may have growth potential, and equipment that can handle larger batch sizes without compromising performance is essential for future expansion. It is crucial to recognize the differences among freeze drying equipment options and select equipment based on specific requirements. Misconception: The same cycle can be used for all products. Reality. Each product requires a customized freeze-drying cycle that considers its unique characteristics and requirements. Optimizing freezing and drying parameters, such as shelf temperature, chamber pressure, drying time, and ramp rates, is crucial to preserve the quality, efficacy, and stability of the product. These parameters are determined based on factors like the product's formulation, physical properties, desired moisture content, and potential degradation mechanisms. To determine the appropriate freeze-drying cycle for a specific product, comprehensive testing and evaluation are typically performed. This iterative process helps in designing a cycle that fulfills the product's specific needs while ensuring optimal drying performance and maintaining product integrity. It is important to seek guidance from freeze-drying experts who can offer valuable advice and assistance in developing suitable freeze-drying cycles for different products. Their expertise ensures that the cycle is tailored to the specific product, resulting in successful and efficient freeze-drying processes. Misconception. The only critical quality attribute is residual moisture. Reality. While residual moisture content is undoubtedly an important attribute, there are several other critical quality attributes that significantly impact the assessment of freeze-dried products' quality and performance. 1. Residual moisture content. The moisture and solvent remaining in the freeze-dried product after the drying process affects stability, shelf life, and reconstitution properties. 2. Product appearance. The visual aspects of the freeze-dried product, such as color, shape, and physical integrity, are crucial for product acceptance and marketability. 3. Reconstitution time and efficiency. The time it takes for the freeze-dried product to fully rehydrate and the efficiency of the reconstitution process influence product usability and experience. 4. Reconstitution uniformity. Ensuring consistent solute dispersion and concentration throughout the rehydrated product is critical for accurate dosing and efficacy. 5. Chemical and biological stability. The stability of chemical and biological properties, including the integrity, potency, and activity of active pharmaceutical ingredients or biological molecules, is vital for maintaining product efficacy and safety. 6. Integrity of container closure system. The container closure system's integrity, including vials, stoppers, and seals, is critical for product protection, sterility maintenance, and prevention of moisture ingress during storage and use. 7. Lyophilization efficiency. The efficiency of the lyophilization process, encompassing factors like drying time, energy consumption, and yield, impacts process scalability, cost-effectiveness, and product quality. All these critical quality attributes collectively contribute to the overall quality, stability, and performance of freeze-dried products. Evaluating and controlling these attributes throughout the freeze-drying process ensures the production of high-quality and reliable freeze-dried products. Misconception. The product is stable after freeze-drying. Reality. Freeze-drying helps enhance the stability of products, but it does not guarantee indefinite stability. Freeze-dried products are generally more stable compared to their non-dried counterparts or products preserved by other methods. However, the stability of a freeze-dried product can be influenced by various factors, including the specific characteristics of the product, formulation, storage conditions, and packaging. Freeze-drying removes moisture from the product, reducing the potential for microbial growth and chemical reactions that can lead to degradation. By removing water, freeze-drying creates a state of low water activity that helps inhibit spoilage and microbial activity. Additionally, freeze-drying can minimize changes in the physical and chemical properties of the product during storage, such as particle aggregation, enzymatic degradation, and oxidation. While freeze-drying provides significant stability advantages, it is important to consider other factors that can affect the long-term stability of a freeze-dried product. These factors include the formulation and composition of the product, compatibility with the chosen excipients, the presence of sensitive active ingredients or molecules, and the selection of appropriate packaging materials and conditions. Proper storage conditions, such as maintaining low temperatures and protecting against moisture, light, and oxygen, are critical for preserving the stability of freeze-dried products. Additionally, routine stability testing is often conducted to monitor and assess the product's stability over time. Misconception. The same container closure system can be used for all products. Reality. Selecting the appropriate container closure system is of utmost importance in freeze drying. The container closure system refers to the combination of the container, vial, ampule, syringe, etc., and the closure, stopper, cap, seal, etc., that houses the freeze-dried product. Here are some key reasons highlighting the importance of selecting the right container closure system. 1. Product protection. The container closure system plays a vital role in protecting the freeze-dried product from external factors such as moisture, oxygen, light, and contaminants. It creates a barrier that helps maintain the integrity, stability, and sterility of the product throughout its shelf life. Proper closure system selection ensures adequate protection against potential degradation or damage. 2. Moisture control. Freeze-dried products are susceptible to moisture absorption, which can compromise their stability and quality. The container closure system should have excellent moisture barrier properties to minimize moisture ingress and maintain the low moisture content achieved during freeze drying. It helps prevent product rehydration, crystal growth, and potential degradation caused by moisture. 3. Gas barrier. In addition to moisture, the container closure system should provide effective gas barrier properties. It helps prevent oxygen and other gases from entering the product, which can lead to oxidation, degradation, or changes in the product's chemical composition. Maintaining a controlled gas environment within the container aids in preserving the stability and efficacy of the freeze-dried product. 4. Sterility and contamination prevention. The container closure system should ensure the sterility of the freeze-dried product. It should provide an airtight seal or closure mechanism that prevents microbial contamination during storage and use. Maintaining a sterile environment is crucial for pharmaceutical and biological products to meet regulatory requirements and ensure patient safety. 5. Compatibility with product and process. The container closure system should be compatible with the freeze-dried product and the freeze-drying process itself. It should not react with the product or introduce impurities that could compromise its quality or stability. Additionally, the closure system should withstand the freeze-drying process conditions, including freezing, primary drying, and secondary drying, without compromising its integrity or performance. 6. Packaging integrity. The container closure system should maintain its integrity throughout the product's shelf life. It should be resistant to cracking, breakage, or leakage that could compromise the product's sterility, stability, or physical integrity. Proper packaging integrity ensures that the freeze-dried product remains protected and preserved until it reaches the end user. Selecting the appropriate container closure system is crucial for ensuring the preservation, stability, sterility, and quality of freeze-dried products. It helps protect against moisture and gas ingress, prevents contamination, and maintains the integrity of the product during storage, transport, and use. Careful consideration of the container closure system is necessary to meet regulatory requirements, ensure patient safety, and deliver high-quality freeze-dried products to the market. Misconception: all water freezes during nucleation. Reality: only about 10% of the water freezes during nucleation. Contrary to a common misunderstanding, freezing is an exothermic event, meaning it generates heat. Water will supercool below the 0 degrees Celsius before it can freeze. In clean environments where there are very few particles to offer nucleation sites, the water can reach -18 C before nucleation occurs. As the water freezes, it warms up to 0 C. Once it reaches 0 C, the nucleation process ends. At this point, only about 10% of the water in a vial will have formed ice crystals. Further cooling is required to fully freeze the water. Therefore, 90% of freezing occurs after nucleation. The nucleation temperature and rate of cooling during the post-nucleation freezing process influences the size of the ice crystals formed. Lower nucleation temperature and faster freezing leads to smaller ice crystals, while higher nucleation temperatures and slower freezing results in larger ice crystals. Larger ice crystals are easier to freeze dry. Misconception: all vials nucleate randomly during the freezing process. Reality: while the initial nucleation event may be random, once vials start nucleating, neighboring vials are inhibited from nucleating. Nucleation is an exothermic event. The temperature of the nucleating vial will increase to 0 C. Since the first vials to nucleate increase in temperature, the 6 vials that surround the nucleating vial are robbed of their cooling. The nucleating vial will need to cool down to a lower temperature before the adjacent vials can nucleate. This process of vials inhibiting adjacent vials can repeat several times during most freezing cycles. Misconception. Controlled nucleation always shortens primary drying times. Reality. While controlled nucleation promotes uniform structure and consistency across the batch, it may not always shorten primary drying times. In cases with small fill or high solid content, controlled nucleation may not impact primary drying time.